Yeah, so let me first thank Arijit and his team for lovely hosting. It has been really a pleasure for last 2 days and enjoying the talks. So I'm going to talk about gene-environment interactions, and this is the title of my talk. And I was first thinking to make it more theoretical work that I have done, but since it's now the closing session, so I thought maybe I will just show some of the applied work that I have done and that we plan to do in the future at IIT Jodhpur. Like this familiar, like this, how the humans have expanded over the years. And this is a lot of population genetic studies have been done in this context, and modern humans they have evolved from Africa and spread across the world, adapting locally to uh, different selective pressures of the climate, food sources, and also pathogens. And on the right side, you see that how humans have successfully colonized nearly in all kinds of habitats, whether it is high altitude or arid regions and different latitudes. And also, there has been a lot of shifts that has happened over the years in the cultural practices of our human existence, and they have been linked to prevalence of the disease. Uh, for instance, the prevalence of dairy farming has been strongly correlated lactose intolerance. So, on the right side, you see that many of these conditions that we think of as disease nowadays, they actually give us our ancestors benefit in the survival and the early stages. So, another thing that is more pertinent to Rajasthan region where IIT Jodhpur is located. So, the domestication of wheat and the prevalence of celiac diseases nowadays, the prevalence of celiac disease in Rajasthan region is increasing because traditionally they have been eating millet-based food, but now they have shifted to the wheat-based. So that probably indicates that kind of change that we are seeing now. But not only this, so there has been a lot of interesting work uh, that has happened across the world actually. And you can see the first work that is very old in 1963, that paper was published in Science, where they have taken the African and Middle East desert reed. They have taken it to the lab and they have taken it to the non-native environment, which has actually resulted into diabetes mellitus in these cell rats. So another work, but somehow this was not followed up by people later, but now in 2017, there was a new study which actually followed up this work, and they showed that genome sequence has revealed that it has a mutation in a gene that allows it to adapt to this high aridity and low calorie intake. So this is one example of how the health transitions are happening nowadays with the humans also. So another interesting thing, stories, studies that have been published. One of them is Nature Immunology paper in 2021, where they have shown that the urbans they are living in healthy environments is associated with an inflammatory status driven by dietary and metabolic changes. So how these transitions, and most of these people who are habituated to rural and kind of the traditional cultures, but they have now migrated. So they have been shown and studied with using multi-omics approaches how this has caused these inflammatory diseases in these populations. And another study which is done in Kenya, which showed the adaptation to water stress and pastoralism in Turkana, which is also in the Kenya desert. So they have identified a lot of genes which have allowed them to adapt to this water stress. These are very nice summary of the genes which are having functional and relevance, and they are allowed to adapt to different kind of stresses. to humans and animals. This is published in Trends in Ecology and Evolution. So these are some of the genes which are known to have allowed us humans to have reduced longitudinal water loss, and for example, this enhanced use of metabolic water and tolerance to high salt diets, etc. So these are some of the studies that have been compiled here, and they have been also replicated independently in other papers as well. So this all why I'm showing you is to show you the importance of gene-environment interactions nowadays, which has been missed out in the last few decades, because when the Genome-Wide Association studies started, people thought that everything, all the variances in the phenotypes could be explained with the help of genotypes or genetic architecture, but that has not turned out to be the fact. So what is gene-environment interaction? That's what I am working on, and I have been also working during my PhD work, and also some focus now at IIT Jodhpur. So let me just first introduce you what I mean by gene-environment interaction. So as we know now that most of these human traits and diseases, they are influenced by how different genes, they interact in a complex way with the environmental factors. Say that gene-environment interactions refers to the modification of the genetic effect by the environment. And this is illustrated in this picture. If you have a look, saying the horizontal axis, we have environmental exposure, which is, for simplicity, has been given the two values like low and high, and on the vertical axis we have the phenotype value. So what you see here is this genotype. So let's say somebody having a genotype A, which is kind of standard notation that is followed in genomics, statistical genomics. So so what you see is that these have a nominal level, it's close to low level of the phenotype. And in contrast, these two other groups of individuals carrying another genotypes, AB and BB, they have a high expressed phenotype value of that particular phenotype. So let's take this one example to illustrate this. So what you see here is two identical twins. They are born with a high fat absorbing genes, let's say, and I'm just calling them just random name, John and Tom. So John is following the healthy diet and lifestyle, whereas Tom is follows unhealthy and lifestyle, maybe like smoking or unhealthy food. So what you see, what we see, if there is an interaction with the environment, that there is less risk of obesity and diabetes in John compared to Tom, who has high risk of obesity and diabetes onset. Probably these genes becomes more activated when you change the environmental exposure. So let me just take you through how we estimate then these environment interactions. So what are the methods? So there are classically designed case-control study design, which I feel has been also exposed to you in previous sessions. So we have the cases of the controls, and we have their genotype data and their environmental information. So let's say in this model, which is very simple actually, which is logistic regression model, let's say D indicates the disease status, which has been assumed to be, for simplicity, binary. 0 means no disease, 1 means having the disease, and similarly a genetic factor and environmental factors, they also have been assumed to be binary. So what you can do is you can fit your genotype and environmental data to this model, and the gamma actually here represents the interaction term. So you can express these coefficients of this model in terms of odds ratios, but just taking the exponentials. So what is our interest here is the gamma. which estimates the interaction between genetic and the environmental factor. So, in this, you can define odds ratio ORG as disease odds ratio of the individuals carrying the genotype relative to those not carrying any of the risk factors, let's say. And similarly, you can define ORE and ORGE. So this is the conventional approach. What is the limitation with this? Obviously, we need the controls, right? So what I have done through my work is I have proposed a new, not a proposed, but it has been already been proposed by Pigos in 1994. Again, this was lost because when the genotype in the North Human Genome Project was completed, everything just was changed because environment was not in the picture at that time. So, I propose I include this proposed case-only design. So the assumption with this design is that these G and E are independent in general in the population, which is most of the time true because a genotype profile doesn't decide to expose a profile. So in general, these assumptions are true and disease is rare. So these are two assumptions that one has to check in order to use this case-only design, which is a non-traditional approach. And now, if these assumptions are true, what you have to do is you just have to check association between G and E in the cases that will actually point towards the gene-environment interaction. So there is a theoretical proof that has been given by this paper. So if anybody is interested in knowing the theory of this proof, then they can have a look at this paper. So simply the model reduces to a simple logistic regression where you have just environment and the genetic information. So just fit your in this reduced model. And interestingly, the estimate of this theta that this proof has been given here gives you the estimate of the gamma in the full model. So now you just have more statistical power also because you have less number of parameters to estimate. So this is the case-only way to estimate the interaction. And the beauty of this model is that we don't need the data on the controls. We just need the data on genotype and exposure of the cases. So a lot of money gets saved. And also there are challenges with the recruiting the controls, a lot of challenges. So these are some of the contributions that I have done in this field. So some of these two theoretical paper where we have checked the role of linkage disequilibrium and how we can correct for population stratification issue in while using such kind of design. On the right side, this is what I plan to show you today. There is a work that was done while I was abroad to study genetic factors that influence, that gets influenced in the presence of tobacco smoke and to modify the risk for inflammatory bowel disease. And the second work that I have just recently got published. So this is the first study that I'm going to show you how you can make use of the case-only approach and identify very interesting interactions. So inflammatory bowel disease is a very prevalent disease in most of the Western European cultures. It is a chronic lifelong illness of early onset. There are two principal types of this disease, Crohn's disease and ulcerative colitis. And a lot of genome-wide association studies have been done to identify more than 200 risk loci that have been associated with the risk of IBD. Unfortunately, as most of the jivas have done, they do not explain most of the heritability of the diseases. As you can see, it's just 13%, 8%. So, environmental factors which are relevant for this disease has also been studied, and one of them is the smoking and of course also the diet. But what we did is we had been lucky to find some data lot of data, some but lot of data which having smoking information for these patients. So this study that I did is multicentric gene smoking interaction study, and it was done in collaboration with the International IBD Genetics Consortium. And luckily, they had a huge amount of data to be used for this case, all this estimation of the interaction. So they have 10,000, more than 10,000 Crohn's disease cases and around 8,000, more than 8,000 ulcerative colitis. And also, they have the genotype data of these patients. So these are all patients and their genotyping data. So what we did is we did a meta-analysis. So we perform center-specific analysis, and then we perform a meta-analysis combining the center-specific studies. On the right side, you see just an example of the data. It's not the full data, just Crohn's disease data from 10 centers. Just to show you that how the data looks like and what kind of data would be required. So you have we have the data from US, which is having the current smoking rate of 11%. On the other hand, in Australia, it was found to be around 44%. So you see that there is also heterogeneity across the centers in terms of the smoking rate in different countries. So this also kind of raises a concern of population stratification for some of the centers, because US and of course also UK, they have a lot of admixture populations. So one of the theoretical work I did was to how we can correct for such kind of biases that can come from population stratification. So I am not presenting that work, but what you see is the use of that work in this work. Here I did the same correction for population stratification on this data, and this is actually done through this genomic inflation factor, and it was estimated to be 1.1 in US, which was expected. So the lambda greater than 1 indicates that there is you have to correct for population stratification. So for those centers having this US and mainly the Australia and UK, we had to do the population stratification correction, and for that, one has to use an independent set of slips to calculate this lambda. So these are some of the interesting findings from this work. So we also did some of the experimental validation with the help of our collaborator in France. What we find is we find that finding from mice and the humans, they both reveal the effect of smoking on the risk of IBD. IBD that would depend on genetic variants. So through all the meta-analysis work that we have done, so we have identified 64 SNPs that show the evidence of gene smoking interaction, and some of them were interestingly also validated in the mice. So these gene smoking interactions were confirmed in the mice with the disruption of IL-10 and known variants of these genes, which have also been associated with the risk of IBD in previous studies. And we also replicated the interaction of a variant in the NOD2 gene with the current smoking in relation to the risk of Crohn's disease, which is also a kind of a validation from what we have been obtained for using the case-only approach. So this showed the validity of this case-only approach, and also show the benefit that you don't have to spend your money on unnecessary money on controls. Yeah, so that was this work here, and now I want to show you some of the work that I have done very recently, and it has got published in scientific reports. So what our hypothesis was here that these cancers, they are driven by, related to type 2 diabetes. So type 2 diabetes acts as a precursor or intermediate. Whereas there is a hypothesis, but we have to kind of little bit change our plan. So what you see here is we had the data from type 2 the type 2 type 2 diabetes patients and cancer patients, and our aim actually was to identify hub biomarkers that are potentially able to predict the chance of cancer onset in the type 2 diabetes patients, which I'm just noting with T2DM. Um, so why it is so? So we know that these two phenotypes, they share share exposures. They have shared exposure like obesity, unhealthy lifestyle, and they have the aging, etc. So they share both exposure or lifestyle in common. So we thought that there should be also some genetic markers that are also common across these phenotypes. So we did our analysis, and we were able to identify 18 such genes which are shared between these two phenotypes. So what we did is we couldn't look all the cancer types because we did some of the literature work and found that epidemiological studies in the past, they have been able to kind of guide us that these three cancer types, they are more relevant in the context of type 2 diabetes. So we identify five genes that are common with the breast cancer, and 6 for the liver cancer, and 7 for the pancreatic cancer. And these are the genes which we have been able to identify from our work. And this work is published in scientific reports. And we have, I'm lucky to have biologist on board, Dr. Priyanka Singh from IIT Jodhpur. And this work was supported by IIT Jodhpur and GI Germany. So let me just take it through what we have done in this study. So this was the data that was publicly available. So as I said, so data was a challenge here to study this work. So we had nothing that is publicly available. So we went to GEO, which is one of the largest openly accessible database. We took up these, we did a lot of search, and we found these 3 data sets to be most suitable for this problem. We don't have other better options. These were the best options we could get. We had patients that are on type 2 diabetes, 8 number of patients, then 8 number of patients who has the pancreatic cancer, and this data actually have also the patients which have both type 2 diabetes and pancreatic cancer, and healthy patients, healthy individuals. Similarly, the other 2 data sets we have taken. And a lot of miscellaneous, unfortunately, we didn't have other options. So this dash actually indicates the miscellaneous. So this is the approach. So what we did is we first did a DAG analysis, differentially expressed genes, comparing healthy samples with the type 2 diabetes. Similarly for the cancers, and then we find the common genes that are coming from these 2 analyses. And then with a lot of stringent criteria to ensure that there are no false positives. So some of them are shown over here, which are familiar probably to you, but we also applied another filtering criteria to ensure that we are getting the right or not the false positives. So then we did some classical standard analysis like functional enrichment analysis, and then pathway analysis, network analysis, and so on to validate our findings. So these are some of the results that I think maybe some of you might be interested to look into. So we have I'm showing you here the type 2 diabetes and the pancreatic cancer results that from the network analysis. So we have been able to identify this 7 of these hub genes you can see over here based on our analysis. And just to summarize what we have found from this analysis, so we have found 7 genes, hub genes from T2D versus PC comparison, 6 from T2DM versus liver cancer, and T2DM versus breast cancer, 5 genes, which are shown over here. And you can see, I think most striking thing here to note is that we have just considered those genes which are expressed in the same direction in both the conditions, which is making sense in the way that we are just interested in those genes which might be causing the further cancer, leading to the cancer in type 2 diabetes patients. We also did some of the survival analysis for some of these genes identified, and said these genes were strongly associated with poor survival in this liver cancer patients. This study is that we were able to provide insights into biological and molecular events that could link T2DM with 3 common cancer types. And our enrichment analysis highlighted the enrichment of cell cycle events, immune system processes, and cell morphogenesis, and gene expression and metabolism as biological processes. And the identified genetic markers, they hold the potential to predict the chances of cancer onset in T2DM patients. This is something, this is what's first kind of work to our knowledge that has been done in this context. And of course, there are a lot of more data is required, and a lot of other functional validation of these is required to kind of corroborate these findings. So this is something that I have done using publicly available data sets. Now I just want to share with you what I am planning to do at IIT Jodhpur, because it has geographically, it has a lot of advantage to perform these kind of studies. So this actually is close to, and Jodhpur is close to Thar ecoregion, which has a lot of climatic stressors like the aridity and high UV, extreme temperature, and so on. And in addition to that, there are a lot of interesting environmental exposures to explore. For example, there are people who are working on craft, they are working in textile, they are painting, leather, stone, etc. So there's huge opportunities to explore how gene-environment interactions could be studied in these populations. So what we want to do is, and this has not been done in past in any of the work. So because this is not been studied, so we first want to study and perform their ancestry analysis to find from where these native populations are actually mapping to, and then we will be able to hopefully identify signatures of selection, just like the Turkana and Tanzania work that has identified. And also, what we are planning to do is identify the populations, native populations, who migrated to urban environments, because a lot of transition is happening, because this happens naturally because of the job requirement, etc. So we want to study how these transitions are actually leading to this non-communicable complex diseases. So we plan to perform multi-omic studies of these Thar populations in native and the transition environments. And of course, there is a huge opportunity to perform genome-wide gene-environment interaction studies for what's called as occupational exposures. This is something which is planned for the future. Just want to share with you, and I want to share with you also some of the very preliminary results that we have got, maybe interesting. And also, in addition to this climatic stressor and the environmental exposure, the food habits are also very unique in the Jodhpur area. So you will see this kind of care and turmeric people are having using it, this kind of food in their routine. So and also interestingly, some of these studies, they have find that they have these food habits. They have significant medicinal properties like cardioprotective, anti-inflammatory, and so on. So how they actually allow them to cope up these kind of diseases. And also one interesting fact, just if you like maybe observe it, that in desert, if camels and most of the animals, they store the food so that they can make use of it when there is a starvation. So also, I think the same kind of phenomena has been also been reported in one of the study where the glucose level of the healthy individuals is more than 200. This is because they are used to such kind of environment that they have to keep their glucose level higher so that they can, that gets to kind of accommodate the starvation conditions. So these very interesting things and phenomena that we plan to explore, and this is what we want to do. So we have a lot of things that we plan to do. So we collect the phenotyping data and genotyping data, and also exposures, and perform comparative genomics, comparative transcriptomics, and etc. So IIT Jaipur actually gives also another benefit, which is very unique, I would say, is the transdisciplinary research that can be enabled, because such kind of work, it's immense work and it requires a lot of transdisciplinary expertise. So at IIT Jaipur we have departments and schools. For example, there are also engineering sciences, like we have electrical, bioscience, engineering, and traditional computer science, and so on. And then there are schools of management and entrepreneurship and school of liberal arts. And in addition to that, there are the IDRPs, which are interdisciplinary platforms for research platforms, which people from different disciplines, they sit together and they can work on the same problem. And also we have School of Artificial Intelligence and Data Science. So given this, there is a huge possibility and make use of this transdisciplinary environment to do such kind of study. And this is a long-term realm that I'm showing you here. So we want to have all these modalities and use it as an input data, and maybe use DL algorithms to in future to guide the virtual health guidance. So this is a long way to go. So just quickly, I will show you one study that we have been done so far very recently. So we have actually done the genotyping of around 185 individuals which belong to these communities and so-called the Thar communities populations. And this work is done in collaboration with the IGIB, and also this identification of the native populations done with the anthropologist Dr. Jha, and this is supported by seed grant from IIT Jodhpur. What we have shown that this integration with the previously available IGV data set, that these populations in the Thar, they are reveals proximity to Indo-European populations. This was actually also suspected because they belongs to geographically in that region. So this is just, I think, the last slide, second last, I guess. So I have shown you 3 studies. Study 1, which was done to study the effect of smoking on the risk of inflammatory bowel diseases, and we have been able to identify 64 genetic variants which are influenced by smoking. Second study I showed you, where we have identified 18 hub genes which link type 2 diabetes with 3 common cancers. In the third one, which is unpublished, of course, and there are native Thar populations which reveal proximity with Indo-European populations. And this is just an announcement. So we are having diploma and certificate program for working professionals in omics. So the next batch will start from July 2024. So if anybody is interested, you please feel free to forward it, or you can talk to me later. That's also fine. So with this, I would like to thank my young team, which so Rajveer has worked on the diabetes paper, And I would like to thank all my collaborators and funding support. Yeah, thank you.
Let's thank Pankaj for this nice talk. Maybe we have time for 2 questions.
So, nice talk, sir. Thank you for the talk. So, my question is, what was the initial spark to hypothesis that diabetes will be related to cancer? And is it like whoever has diabetes should undergo such testing for cancer? Because you told there are factors like obesity and things which are common to both the things. But this obesity, unhealthy lifestyle can also be related to other whole range of metabolic disorders, heart diseases, etc.
That's true. Yeah, so I think that, as I said, so we did a literature review to first identify what has been studied. So one cohort study actually in China that have been done for 10 years, where they have followed the type 2 diabetes patients, and then they checked the progression of the other phenotypes. So like those kind of studies, and of course there are, I think, I don't think there are mechanistic models that have been studied so far, but this is something I think people should be doing in future now based on these findings.
And also, sir, have you taken into account the batch effects while you're taking data from GEO databases?
Yeah, so that's good questions, and we are independently analyzing it. So we are not merging it. So that's the batch effect doesn't come into picture here. But if you take for the same phenotype the other data set, then it will be problem.
Nice talk, Pankaj. Just curious to know that how did you define the hub genes in your first work that you presented?
Yeah, so in the method analysis part, there are a lot of features that are calculated. I think 14 or so different features, topological features, and based on that, one can identify how that particular gene is connected with other genes, and what are the other topological features. So based on that, they have been identified.
So is it like RNA-seq co-expression network? That's what you were telling?
It's the gene expression data. Something like that.
So these genes are not the genes that are implicated in T2D GWAS or anything. It's pure, you're starting from the gene expression itself.
Yes. So unfortunately, we didn't see much of those that have been identified in GWAS. We looked at those, specifically those genes also, but our study didn't link to those. I think there are 3 or 4 main genes that have been identified for type 2 diabetes.
Okay, so then how did you start with how did you zero in on these genes? I'm not talking hub genes, but then how did you zero in on these genes? Is my question clear?
Not really. Maybe you can explain it, or maybe we can talk later.
Okay, sure. And what was the contribution from PC1 and PC2 in your last study?
Not much. This is very recent work I am showing you. It is very less, because the sample size is also very small, under 85. So we plan to also merge it with maybe larger samples and check it.
Did you say that GSA has 7 million markers?
Yes, I think so. It should have. It may be. It may be, yes, 7 million on NOAA-seq. I think it is. It is less, I think. Yeah.